There are many antimicrobial agents whose mode of action is protein synthesis inhibition. So let us look at some of these protein synthesis inhibitors. These protein synthesis inhibitors, they inhibit the process of protein synthesis or translation in bacteria. So let us first look at tetracycline. Tetracycline interferes with the attachment of tRNA to mRNA ribosome complex. So as we know that in translation we have the mRNA, this is the mRNA, and this is the ribosome, the 30th part of the ribosome and the 50th subunit of the ribosome. So the tRNA that brings the tRNA which carries the new amino acid, the tRNA that is carrying the new amino acid which has to be incorporated into the polypeptide chain. The tetracycline interferes with the attachment of this tRNA to the mRNA and ribosome complex. This is the mRNA ribosome complex. So the tRNA has to come and attach. So this attachment of the tRNA is inhibited by tetracycline. So no new tRNA can attach to the mRNA and ribosome complex. So the polypeptide synthesis is inhibited. So this is the mode of action of tetracycline. Now let us look at the mode of action of chloramphenicol. So what chloramphenicol does it binds to the 50th subunit of the ribosome. It binds to the 50th subunit of ribosome and it inhibits the peptide bond formation. So in the end, a peptide bond has to be formed, right? This is the new amino acid that has to be incorporated, and this is the already existing polypeptide chain, and a peptide bond has to be formed. This amino acid has to form a peptide bond so that it also gets incorporated into this polypeptide chain. So this peptide bond formation is inhibited by chloramphenicol. So chloramphenicol inhibits this peptide bond formation. So there is no peptide bond formation, the polypeptide chain doesn't get elongated and protein synthesis is inhibited. So this is the mode of action of chloramphenicol. Now let us look at the mode of action of streptomycin. So streptomycin, it binds to the 30th subunit of the ribosome and it distorts the shape of this region. It distorts the shape so that the code on the mRNA can't be read correctly. So as we know that the mRNA, they have codons, which is the code which code for a specific amino acid. So because of this distortion in the shape, the code is read incorrectly, right? It is not read properly and an incorrect amino acid is incorporated. So an incorrect amino acid gets incorporated and the protein is faulty. A faulty protein is synthesized. It is not functional, right? So as it is not functional, it is not beneficial to the host. So this is how streptomycin inhibits protein synthesis. So these are three different drugs: streptomycin, chloramphenicol, tetracycline, which affect different parts of the translation process and inhibit protein synthesis.